All right, we're going to continue on with chapter 16. This is the last topic of our unit. It's kind of a short unit. We've talked about embryo development, and we've talked about cancer. Now we're going to talk about stem cells. So remember, the overarching theme here is cell division and gene regulation. And so I want to start with cloning. If in organismal cloning, one or more organisms develops from a single cell without meiosis or fertilization, so without the usual reproductive procedure, the cloned individuals are genetically identical to the parent that donated the single cell. So the thing being cloned, the clone is genetically, their DNA, the base sequences of DNA are identical. And cloning is of interest because of its potential to generate stem cells. And so stem cells, remember, are cells that are undifferentiated, that have no job, that can be manipulated to form into certain types of tissues or certain organs. And so the way that animals have been cloned is this process called nuclear transplantation. So in the cloning of animals, the nucleus of an unfertilized egg or zygote is taken out, and then the nucleus of the cell that you want to clone is put in. So you would take the nucleus of the thing you want to have cloned, one of its diploid cells, the nucleus that has all of the chromosomes in it, and you'd put it into an egg. Because remember, the egg has instructions. And then that egg would develop into the cloned individual. And so with frog embryos, so in order to clone frog embryos, they did nuclear transplantation, this guy named John Gurdon. And what he found with frogs is that the older the donor cell, the less successful it is. So the older the donor nucleus, the lower percentage of normally developing tadpoles. And so nuclear transplantation is restricted as development and differentiation proceeds. And this picture is just showing you what happened with the frogs that he studied. On the left side, he's cloning an embryo, taking the nucleus out of an embryo, putting it into an egg, cloning it into another embryo. So what you end up with are two tadpoles that are genetically identical. Then on this other side, he took a tadpole that had already, so a frog that already developed into a tadpole, cloned the tadpole using one of its cells, and then it didn't develop into another tadpole. So he found that he could only clone embryos. He couldn't clone tadpoles because as the frog got older, its cells were differentiated and more differentiated, and it was too difficult to actually clone a successful tadpole. However, there have been reproductive cloning of mammals, like Dolly the sheep. So in 1997, Scottish researchers announced the birth of Dolly, a lamb that was cloned from an adult sheep, which is like crazy. So they took one of the mammary cells of this adult sheep, took its nucleus out, put it into an egg, and then did like in vitro fertilization into a sheep, and then Dolly grew inside the uterus of a sheep and was born in 1997. The problem is that she had a premature death. She had arthritis. I know she had a lot of lung issues, and it was just kind of noted that she wasn't as healthy as a normal sheep. And so what happens is the donor nucleus needs to be reprogrammed or deprogrammed almost completely because there's epigenetic changes that can lead to health problems in the clone. Part of the reason why human cloning is not legal is it's unethical, since the other mammals that have been cloned have had so many problems. And there's a picture of Dolly and the donor. So she's the clone of the donor. And so I'm going to talk a little bit about how they don't look identical. So cloning has been demonstrated in mice, cows, horses, pigs, dogs, and monkeys. There's a cat called Carbon Copy. This cat was cloned at Texas A&M University in 2001. So in December of 2001, a calico female cat was cloned from another calico female cat. And so the clone was called Carbon Copy. The parent, or the donor, her name was Rainbow. And so what was found is that, so this is Rainbow on the left side, and this is carbon copy right here. So carbon copy is the clone of Rainbow. What you'll notice is they look different. So cloned animals do not always look or behave exactly like their parent. So kind of cool that carbon copy was cloned at Texas A&M University. The other thing that was really cool is that carbon copy did not have any health problems. She lived for 19 years. She just passed away on March 3rd of 2020. So right before we left for spring break, she passed away, which is kind of sad, but kind of cool that she lived for so long. And so with nuclear transplantation, only a small percentage of the cloned embryos actually develop to birth. Most of the cloned animals exhibit defects, kind of like Dolly the sheep did, and it's believed that the epigenetic changes in the donor nucleus really need to be successfully reversed and completely reversed in order for the genes to be expressed or repressed appropriately. So it's believed whenever these cloned mammals had a lot of health problems, it's because the donor nucleus was not completely deprogrammed, all right, and the epigenetic changes. Remember we talked about methylation and acetylation that happens to DNA. Those have to be successfully reversed in the donor nucleus. And scientists just have not perfected that yet. There's other reasons why humans, like, it's not legal to clone humans, but that's one of them, is that it just, you would purposely make a human that would have problems, and that's just considered unethical. So I have a video that I'm going to show you next time I see you about if you want your dog cloned, how you can send tissue over to a laboratory and get your dog cloned. But the problem is they can end up with some major health issues. So it isn't actually an appealing thing to do, although it is a business. And so I'll show you that video next time I see you. We're going to switch gears here and we're going to talk about stem cells. So you're going to want to write down this definition. A stem cell is a relatively unspecialized cell that can reproduce itself indefinitely. And so it's not going to have a job. So it's completely undifferentiated and not have a job yet. So unspecialized, the genes have not been turned on and off yet. We have different types of stem cells depending on the age of them. So totipotent, so what you're going to write on that left-hand side is a totipotent cell is a cell that comes from an embryo that's only 1 to 3 days old. A totipotent stem cell can actually develop into a new individual. So it's basically like a real clean slate. So 1 to 3 day old embryos. Pluripotent stem cells come from an embryo, or it's also called a blastocyst, that's 5 to 14 days old. And these are the ones of interest that can be grown into different tissue types. And then multipotent stem cells can come from fetal tissue, cord blood, or even adult cells. Unfortunately, multipotent stem cells can only differentiate into fat, bone, or blood. The things that we aren't really interested in growing from stem cells. Okay, so they come from fetal tissue, cord blood, or adult cells, but they can only grow into fat, blood, or bone. So the ones of interest are the pluripotent cells. And so the question at the bottom of 188 is, when does an embryo no longer have pluripotent stem cells? You're going to write 14 days. So after 14 days, those stem cells really are not of good use. All right, and so what this is just showing you is that an adult stem cell, we have stem cells. An adult stem cell can really only grow into fat, bone, or blood cells. And so that's not quite what we're interested in or what scientists are interested in. All right. So many early animal embryos contain stem cells able to differentiate into any cell type. So remember, that's pluripotent. An embryo between 5 and 14 days. These can reproduce indefinitely. The adult body also has stem cells, which can replace non-reproducing specialized cells as needed, like fat, bone, or white blood cells. But I'm going to talk about how adult body cells actually can become like embryonic stem cells. I'm going to tell you what that process is in just a second. And so what this picture is showing you is an embryonic stem cell could become liver nerve, or blood. And so it's pluripotent. It could become like over 200 different types of specialized cells. An adult stem cell is very limited. So that's all this picture is really showing you, is that an embryo stem cell, it has a lot of potential. An adult stem cell does not. And so that is where the controversy comes into play, is if you take stem cells from an embryo, the embryo then dies. And so that is controversial. So what's really cool is that researchers are trying to overcome that controversy, not by trying to make new laws or change people's minds. They're trying to find another way. How can we make everyone happy by not killing embryos, basically? And so it's this thing called induced pluripotent stem cells. And so what they're able to do is they're able to take an adult cell and deprogram it to turn it into an embryo stem cell. Super cool. And what they're doing is they're using viruses to do that. All right. And so they're using what are called retroviruses. And when we get to our virus unit, I'll talk more about what's the difference between a regular virus and a retrovirus. All I really want you to know is that it's just a virus that's carrying a gene. And what that gene does is it reprograms that cell completely so that those cells can then be turned into embryonic stem cells and then grown into certain types of tissues. And so what this is just showing you, it's from the book, it's an image from the book, and it's just showing how an adult skin cell can have this virus introduced to it, and the virus is carrying stem cell master regulatory genes. And what those regulatory genes do is it integrates itself into the DNA and basically just shuts off the jobs. Like, you are a clean slate. It completely like deprograms it, turns it into a pluripotent stem cell. And so cells of patients suffering from certain diseases can be reprogrammed into induced pluripotent cells, stem cells, for the use of studying the disease and for potential treatments. In the field of regenerative medicine, a patient's own cells might be reprogrammed into induced pluripotent stem cells to replace non-functional or diseased cells. So I will tell you which diseases is this being used for. It's being used for diabetes, and it's also being used for eye diseases, so to replace cells of the retina. And so what's happened is if a person's pancreas isn't working correctly, cells will be extracted from the person's pancreas. They will be introduced with this virus. The virus deprograms it. And then the cell gets reprogrammed and then put back into the pancreas. And then hopefully it starts producing insulin. So type 1 diabetes, this is supposed to be something that has been shown to be successful. Very, 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 very expensive, $10,000 for a treatment, and it's not covered by insurance yet that I know of. And then also used for retina diseases or eye diseases. So the advantages of it, I'll just kind of show you the picture. And you have a picture on page 190. The advantages is that it's easy to use, reproducible, good efficiency, and you can get controlled expression. And so it's done in the lab, then put into the patient. The disadvantages is that it can risk more mutations and can cause incomplete silencing of genes and can just kind of cause more harm than good, basically, make the disease state worse. You can imagine why. And so the pros and cons that I put on page 190 at the very bottom, the pro of IPS is that it would be the patient's own cells that are being reprogrammed. So you don't have to worry about rejection of the tissue. You don't need an embryo. And then the cons is that the cells would still have genetic defects. It could cause cancer, and then the virus might insert genes in places that we don't want them, causing more mutations. So this has been shown to be, like I said, effective in treating diabetes, but still a lot more research needs to be done. So I hope you found some of this interesting. That's really all that I had for cloning and stem cells. Let me know if you have any questions. I'll talk to you later.